So our second speaker today is Xingui Liu, who is currently a Marie Curie Fellow in Professor Alessio Ciulli's lab at the Center for Targeted Protein Degradation at Dundee. And she is focusing her research on developing PROTAC degraders for LRRK2, a promising target for Parkinson's disease. Before she joined the Ciulli group, she was a postdoc at the University of Florida, working with Professor Zheng and Zhao. At the University of Florida, she was part of a team that developed very effective BCL-XL and BCL2 PROTAC degrader molecules, one of which was licensed. She completed her PhD in Dr. Zheng's lab at the University of Arkansas for Medical Sciences, where she designed, synthesized, and characterized delta. Thank you, radioprotectors. I'll hand over to you.
Yeah, thank you, Catherine, for the nice introduction. It's a great honor to be here in this fantastic webinar. So today I'd like to share a project that has been a collaborative project between Alessio Ciulli's group and Dario Alessio's group. It's about the discovery of XL01126, a PROTAC degrader of leucine-rich repeat kinase 2. We also call it LRRK2 or RRK2. RRK2 is a large multi-domain protein. It is involved in familial Parkinson's disease and sporadic Parkinson's disease through mutations in the GTPase domain and the kinase domain. For example, the G2019S mutation, which is the most common RRK2 mutations. And the mutations in these two domains can lead to the increased kinase activity of RRK2, and therefore lead to the neurotoxicity effect, and is also associated with increased risk for Parkinson's disease. And previous studies have found that if you inhibit the RRK2 kinase activity with RRK2 kinase inhibitors, you get neuroprotective effect. So these early observations provided strong rationale to target RRK2 with RRK2 kinase inhibitors, including both type 1 kinase inhibitors and type 2 kinase inhibitors. And DNL151 and DNL201 are the two most developed RRK2 kinase inhibitors that are currently in clinical trials. They are co-developed by Dana and Belgium. RRK2 dimerization inhibitors and GTPase inhibitors have also been proposed to target the RRK2 kinase activity, because RRK2 dimerization and the GTPase mutations can also increase the RRK2 kinase activity. So new modalities have also been emerging that can remove the protein as a whole, including antisense oligonucleotide and the PROTAC. Antisense oligonucleotide is a single-strand DNA that combines to the mRNA of RRK2 and therefore reduce the intracellular RRK2 protein level at the translational level. BIIBO94 is ASO developed by Ionis and Belgium, and it is currently in phase 1 clinical trial. While ASO can reduce the intracellular RRK2 protein level at the translational level, the PROTAC molecules offers a strategy to reduce the protein level at the post-translational level, and it's a small molecule modality. So our lab has been collaborating with Dario's group for developing RRK2 PROTACs since 2018. The first generation RRK2 PROTACs were prepared by tethering HG-10-102-01, which is a RRK2 kinase inhibitor developed in Greece lab in collaboration with Alessio's group. So we prepared the first generation PROTACs by tethering this RRK2 kinase inhibitor with 3 different E3 ligand that are called CIAP ligand, CRBN ligand, also called, and we use the thalidomide, and the VHL ligand. And we built a small compound library and we screened them. And we found 3 VHL-based PROTAC called SD75, SD82, and SD100 showed moderate RRK2 degradation. So we decided to move this project further by further modify this compound. So the second generation PROTACs aims to further improve the potency. So we used the first generation PROTACs as our lead compounds. And given the modular nature of the PROTACs, so our modification strategy focusing on modifying the warhead part, the virtual ligand part, and the linker moiety part. So for the warhead modification, we tried to replace the chlorine with trifluoromethyl group to improve the binding affinity to ROCK2. And we also tried to remove this piperazine part to try to decrease the molecular size of the molecules. And for the virtual part modification, we tried to improve the virtual binding affinity by introducing a methyl group at the benzylic position or fluorine on the phenol group. And for the linker part modification, we tried to change the linker length, the linker rigidity, and the orientation of the linkers. So we managed to obtain the second generation PROTAC library, and then we screened all these compounds on mouse embryonic fibroblast cells, also called MAFs. So we used both wild-type MAFs and ROCK2 G2019S mutant MAFs. So we treated these two cell lines with our compounds or DMSO, using DMSO as control, and the concentration we used was 33 nanomolar and 1 micromolar, and the treatment time was 4 hours. And then using Western blots, we determined the protein level of ROCK2 total, phospho-ROCK2 at serine 935, which is a biomarker of ROCK2 kinase inhibition, and also a phospho-ROCK10 at threonine 73, which is a physiological, ROCK10 is a physiological substrate of ROCK2. And we used the ROCK10 total and tubulin as control proteins. And then we quantified the Western blots, and we found that at 1 micromolar, as shown by the red bars, multiple compounds showed more than moderate ROCK2 degradation, indicating the structure modification strategy was successful. And at 33 nanomolar, we found 2 compounds stand out, XL01126 and XL01134. They are the top compounds that degraded ROCK2 at 33 nanomolar in both wild-type MAFs and G2019S MAFs. And corresponding to the potent ROCK2 degradation, these 2 compounds also show the most potent ROCK10 dephosphorylation. So it seems apparent to us that these 2 compounds are the top 2 compounds in the second generation PROTACs. But when we look closer at them, so structurally, these 2 compounds share very structural similarity. And with XL01134 has a 6 cyclohexyl ring in the linker, and XL01126 has a trans cyclohexyl ring in the linker. And they're also different from the first generation compound in the linker as well. So we want to further profile these compounds in order to choose the best compound to move forward. So we first did the dose-dependent ROCK2 degradation assay using Western blots, and in both wild-type MAFs and G2019S MAFs. So we found SD75 from the first generation PROTACs dose-dependently degraded ROCK2, as shown by the green curves in both wild-type MAFs and G2019S MAFs, but the degradation was only partial in both cell lines. XL01134, shown by the red curves from the second generation, showed much more potent ROCK2 degradation in both cell lines. But this compound showed a hook effect at higher concentration. And XL01126, in contrast, also showed very potent degradation, as shown by the low DC50 values shown here, and the degradation was also more complete in both cell lines, with Dmax at 82% in wild-type MAFs and 90% in G2019S MAFs. So XL01126 induced the most potent and complete ROCK2 degradation among these 3 compounds. And then we did the time-dependent ROCK2 degradation in wild-type MAFs and G2019S MAFs. And we found all these 3 compounds showed time-dependent ROCK2 degradation from the experiment time point from 50 minutes up to 48 hours. But XL01126 showed the fastest ROCK2 degradation, as shown by its much shorter degradation half-lives in wild-type maps and G2019 maps, with degradation half-life at 0.6 hours, which is 36 minutes in GS maps. And it also showed the most complete ROCK2 degradation in the experimental time period. And the max reaches 82 to 92% in wild-type and G2019S maps separately. So XL01126 is the fastest and the most complete degrader among these three compounds. So then the question comes as to why XL01126 with such minor structure difference, then XL01234 showed more better degradation profile. And we know PROTAC has to engage with E3 ligase and the target protein separately before forming ternary complex. So we want to first check if it's due to the binary binding affinity to the E3 ligase. So we did the binary binding to virtual E3 ligase using FB assay. And using virtual 101 was used as a control. And as expected, this compound showed the best binding affinity to virtual. The cis compound, the cis XL01126, which is a non-degrader control of XL01126, has a flipped hydroxy group in the hydroxyproline ring, did not bind to virtual E3 ligase as anticipated, because the chirality of this hydroxy group is critical for binding to virtual E3 ligase. However, we found XL01126, although it is the most potent degrader of ROCK2, showed the least binding to virtual E3 ligase. And then we did the binary binding affinity to ROCK2 as well. And again, we found XL01126 showed the lowest binding affinity to ROCK2 as well. So these two sets of data suggest that our binary binding affinities are not indicative for target protein degradation. So we know after binary E3 ligase and target protein engagement, the PROTAC has to form a ternary complex with E3 ligase and target protein and potentially induce protein-protein interaction. And this can be reflected in the ternary binding affinity and cooperativity. So we did the so we determined the ternary binding affinities and cooperativity of our compounds using ROCK2 engagement assay. So the blue curve is the binding of the compound to ROCK2 in the absence of virtual, and the red curve is the binding of the compound to ROCK2 in the presence of virtual E3 ligase. And you can see the shift of the curve from the blue to the red, indicating a positive cooperativity. And the cooperativity was calculated to be 5.7. However, when we do the same assay for XL01134 and SD75, we found XL01134 showed a much lower cooperativity, and SD75 showed a negative cooperativity. And among all these three compounds, XL01126 also showed the highest ternary binding affinity to ROCK2. So the high ternary binding affinity and the positive cooperativity of XL01126 may explain the better degradation profile exhibited by this compound. So after ternary complex formation, the ubiquitin proteasome system will kick in to ubiquitinate the target protein, and the ubiquitinated target protein will be delivered to the proteasome to be degraded. And in this ubiquitin proteasome pathway, it can be blocked by the treatment of E3 ligase, the Leucine inhibitor, and the proteasome inhibitor. So we did the mechanism study in G2019S maps. And as expected, this compound at 300 nanomolar 4-hour treatment degraded ROCK2 potently as expected. But when we pre-treat the cells with E3 ligase or the methylation inhibitor or the proteasome inhibitor, the degradation was blocked, indicating XL01126 mediated ROCK2 degradation is through this ubiquitinating proteasome pathway. But we found this XL01126 induced ROCK2 dephosphorylation, and ROCK10 dephosphorylation was only partially rescued by the treatment of virtual E3 ligase and methylation inhibitors and the proteasome inhibitors. That is because this PROTAC is derived from the kinase inhibitor, and it still keeps the kinase inhibition activity, which cannot be blocked by the treatment of these two compounds. And in corresponding to the addition of the methylation inhibitor and proteasome inhibitor, we can see the proteasome ubiquitin level was decreased and increased separately. So after all this time-dependent, dose-dependent, and mechanism study, we decided to move XL01126 forward. So we tested if this compound can degrade not just the mouse ROCK2, but also human ROCK2. So we did the degradation assay on the human cell line called peripheral blood mononuclear cells, which is derived from healthy human donors. And we use the cis XL01126 as a control compound. And we treat the cells with these both compounds at concentrations ranging from 3 nanomolar to 3 macromolar. And we then monitored all these protein levels. And we found, as expected, 6XL01126 could not degrade ROCK2 at the concentration we used. At XL01126 showed a dose-dependent degradation of ROCK2 in this cell line, with DC50 at 72 nanomolar and Dmax at 83%. And thanks to this potent degradation, XL01126 showed more than 40 fold improvement in inhibiting the ROCK10 phosphorylation. So to check the degradation selectivity of this compound on the proteomic level, we performed the mass spectrometry assay, or wild-type maps. So we treat this cell line with our compound at 300 nanomolar for 4 hours, and then we take the cell line and label them with take the cell lysate and label them with TMT. And we found out of the 8,000 protein we profiled, we found ROCK2 was the only protein that was significantly degraded, indicating the selective degradation induced by XL01126. And we also found a PDE60 showed up as a lead off target of this compound. So to further profile this compound as an in vivo shootable degrader, we performed a PK study in mice. So we treat the mice with our compound through IV, IP, or PO at dosage of 5, 30, and 30 milligrams per kilogram separately. And then we monitor the plasma concentrations from 50 minutes up to 8 hours. And we found after all this administration, the plasma XL01126 has a high plasma concentration. And after oral dosing, you can see that the compound reached Cmax within 2 hours, indicating a fast absorption phase. And the compound concentration remained at a high level, indicating the metabolism was slow. And eventually the oral bioavailability was calculated to be 15%, which is moderate, but is amazing given that this molecule is given the large molecular size and high molecular weight of this compound. And what surprised us is that we also detect the compound in the brain tissue and in the cerebral spinal fluid, indicating the compound can actually penetrate the blood-brain barrier. So to summarize the talk today, so through PROTAC design, synthesis, and screening, we discovered a virtual-based ROCK2 PROTAC. This compound can induce cooperative ternary complex with ROCK2 and virtual, and therefore induce fast, potent, and selective ROCK2 degradation on the cell line. And in our in vivo PK study, we found this compound is orally bioavailable, and can penetrate blood-brain barrier. So we are still working on this project, and for the future direction, we would like to see if the compound can degrade ROCK2 in vivo, and we would like to profile the safety and efficacy in vivo degradation. And we're also modifying the compound to get a potentially a better preclinical candidate. And we're also working on solving the extremely complex structure of the compound to enable structural-based PROTAC design. So with that, I would like to thank everyone that has been involved in this project, especially my advisor, Alessio Ciulli, and our collaborator, Dario Alessio, who for being a really great advisors and mentors, and thanks them for their enthusiastic help and support. And I would like to thank my co-worker, Alexia, who has been a great working partner. This project will not be successful without our day-to-day teamwork. Yeah, and I really appreciate all the work we have done together. Yeah, she's now a scientist in therapeutics. And I would like to thank all the support I got from the department and from both BCDD and MRC. And I also like to thank the financial support from Owner Pharmaceutics and UKRI. And lastly, thanks for your attention.